This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
Welcome to A Conversation with History. I'm Harry Chrysler of the Institute of International Studies. Our guest today is Professor Carol Greider, who is the Daniel Nathans Professor and Director of Molecular Biology and Genetics at Johns Hopkins University Medical School. She won the 2009 Nobel Prize for Physiology of Medicine, along with Elizabeth Blackburn and Jack W. Szostak. Professor Greider is the 2014 Hitchcock Lecturer at Berkeley. In 1885, the Hitchcock Endowment Fund was established from a bequest made by Dr. Charles M. Hitchcock to institute a professorship at the University of California for free lectures upon scientific and practical subjects. Professor Greider, welcome back to Berkeley.
Thank you. It's nice to be here.
Where were you born and raised?
I was born in actually San Diego, California, but I was raised in Davis, California.
And looking back, how do you think your parents shaped your thinking about the world?
Well, my father was a physicist at UC Davis, and he very much encouraged me and my brother to be able to follow what we were interested in and to try and keep all doors open, and very much encouraged us to be able to explore things that interested us. And so I think that really shaped how I thought about a career.
And your mother also had a PhD from Berkeley. Both your parents had PhDs from Berkeley.
Both my parents had PhDs from Berkeley, although my mother died when I was young, and so it was my father that raised us. And so his influence on sort of where I went was what I remember most.
And was there a lot of talk at the dinner table about scientific matters?
Sometimes, usually about physics, which was a little bit above my head, but certainly it was clear that being able to pursue what you were interested in was a goal. And
because of your father's role as a professor, part of your early years was spent abroad.
Yes, he was on sabbatical at the Max Planck Institute in Heidelberg when I was 12. So I went to Germany and was put into the German gymnasium, and as a consequence learned to speak German either the hard way or the easy way, depending on how you think about it, just being thrown in and immersed.
In biographical material I've read about you, you had an older brother, mm-hmm. and your relationship with him was very important, and both of you developed a sense of autonomy and independence, even by walking to school.
Yeah, walking to school when we were also when we were young, but also in Germany, and being able to get around on our own through the public transportation when we were, 12 and 13. It was important.
So were thinking skills, critical thinking, just something you absorbed with the atmosphere? A physics professor, father, studying abroad even as a young person?
I never really thought of it that way so much. I didn't think of myself as somebody that was particularly good at critical thinking. I was just trying to follow things I was interested in.
And in your early years, you had dyslexia, right? And so you had to develop, draw on this independence and autonomy to figure out how to really cope to make up for what you couldn't do with things that you could do very well.
Well, it wasn't clear what dyslexia was back then, and so all I knew was that, I had to be taken out of my regular classroom for this remedial spelling, and that I had a lot of trouble in spelling, and it wasn't really defined as dyslexia, and so it wasn't until I was a little bit older and looking back that it was clear that was dyslexia. And so I think that's part of what contributed to my feeling like, maybe I wasn't as bright as some of the other kids were, but if I worked hard, I could still do what I wanted. And so that was sort of the image that I had of myself. It was embarrassing to be sitting in grade school and have a special teacher come in and take you out of the classroom in front of all of the other kids.
But you learn to compensate, really, and develop coping strategies and skills.
Later on, they ended up being very useful. I mean, I remember in my high school biology when we had a lot of memorization of anatomy. We were doing anatomy of the cat, and I found it very easy to memorize things. And I realized retrospectively that that was probably because when I saw a word, I couldn't sound out the word, and so instead, I would memorize it. And so that developed those memory skills that ended up having advantages in other settings.
Did you have any teachers before you went to college that helped further what was to become your interest in science?
I was very interested in biology in high school. It wasn't necessarily something that I said I wanted to have a career in, and I wasn't one of the kids that did science fairs or had a chemistry set. But when I was a senior in high school, I thought biology was interesting, and I was interested particularly in marine biology, which is what prompted me then to choose UC Santa Barbara as the place to go for my undergraduate work.
And for you, Santa Barbara was a special place because of a special college within Santa Barbara. Talk a little about that, because it seems that you really developed some important skills there.
Yeah, so in addition to applying to the University of California at Santa Barbara, I also applied to the College of Creative Studies. And I knew about this because there was a professor there, B. Sweeney, who had been a colleague of my mother's, and my father still kept in contact with the family. And she was a professor of biology within this integrated college. So there's literature, science, math, arts, within a small group. And that was particularly wonderful because there was a lot of individual attention. Each student had an assigned advisor, and you could develop a curriculum with that advisor without necessarily having to take certain blocks of classes as the rest of the university did. And I found that very wonderful because it allowed me to be able to do experiments in laboratories earlier than I otherwise would.
And you really got into the science and doing the experiments across different areas. Talk a little about that.
Yeah, so I remember when B. Sweeney, who was my advisor, said to me as a freshman, she really felt that I should be working in a laboratory. And I remember being thrown in as a freshman and having all of these college classes, and again thinking that I wasn't as smart as all of my other colleagues, and I thought, well, that would be overwhelming. And she said, I think you should try it anyway. And so I did, and I started off in laboratory doing some ecology, beach ecology, and I quickly learned that although I was interested in biology, that area of biology didn't really fit my my way of thinking. It was more statistics. And so then through B. Sweeney and interactions with her, she was able to help me get into several different laboratories at Santa Barbara. And it was when I got into a laboratory that was doing more biochemistry, where one could really look at what we now call sort of mechanistic interactions, how you make a change in a system and then you can see the outcome of that change. That really fit, my intellectual way of thinking. So that's was really a wonderful opportunity to test out different areas, because sometimes you can't know what is most interesting to you until you're actually in the environment.
And so it was a matter of self-discovery, what really worked for you. And interestingly enough, it sounded like you wanted a physics dimension to the biology.
Well, certainly a physical dimension.
Physical,
yes.
Yeah. But that's what physicists do, right? Yeah. So that's fascinating. But the structure, the curriculum structure, and the mentoring sort of helped you find what it is you actually wanted to do. That's rare sometimes in undergraduate education.
Yes, and it also helped because I also took a year abroad. Because I already spoke German, I was able to take a year abroad at Santa Barbara and go and studying in Göttingen. So it was my second year abroad then. And it allowed me then to take the courses there and fit them into my academic program. So in taking a year abroad, I still graduated in 4 years.
There sounds like a lot in your experience that made you observant of people and different kinds of people in a way. Is that fair? I mean, to be immersed in a foreign environment at a young age and then doing it again, it must have helped make you an observant person of the differences out there.
Yes, I mean, I think that is certainly something that I was always aware of is, people and interactions with people. And so I think that because I liked that, I liked going abroad, and the going abroad then helped me learn to do that as well.
Now, then after getting your BA, you came to Berkeley as a graduate student. Talk about choosing Berkeley and why you wanted to be here.
Well, when I was applying to various graduate schools, again, one has to take standardized tests, and because of my dyslexia, first for the GREs, for the SATs, and also for the GREs, I didn't do so well on those. And so of the 13 places that I applied to, only 2 considered me for the graduate program, and one was Caltech and the other was UC Berkeley. My advisor, B. Sweeney, at the time, she said, "Oh, you just want to go to Berkeley because that's where your mother went." And she advised me instead to go to Caltech to do something different. But when I came to interview, I was very captivated by the program, and specifically my interactions with Elizabeth Blackburn during my interview here. And that's what really swayed me to come here rather than Caltech.
And what were the years that you were at Berkeley, and what was Berkeley like that just atmospherically in general?
So I was here from '83 to '88, and it was a wonderful, open, intellectual environment, and I very much liked the interactions with my classmates in the molecular biology department. It was people really were interested in what other people were working on, and friends would talk not just about what was going on at socially, but mostly it was about experiments, and there was a lot of support amongst the group, and so today many of my best friends are still the students that I was here with at Berkeley during my PhD.
I like to ask my guests what they see as both the skill set and the temperament that are important for the kind of work you do.
What they would see, or
what you looking back at your own career and doing the work that you do, what is the skill set involved?
Being able to have the freedom to do something that one's interested in without necessarily worrying about obstacles. And I think that that's something that is different today than it used to be. People are a little bit more worried these days about what are they going to do for their career or what might get in the way. But I certainly felt as a graduate student that I just wanted to do what was interesting, and I didn't really see that there were limitations or obstacles. Now, that may be unique to me because I learned early on, perhaps through the dyslexia, to just put blinders on, and when I saw something I couldn't do, just to ignore the fact that I couldn't do it. So it's not clear to me if I was unique in that setting, but certainly I felt that I was extremely encouraged to follow what was interesting and not worry about any consequences.
And you had some choices once you were here in terms of what you would specialize in. What was your thought processes like? Had your experience at Santa Barbara sort of pointed the way, so to speak, in the choices you made there?
I knew from Santa Barbara that I liked this sort of mechanistic way of thinking, and that was in both biochemistry as well as in molecular biology. But conversations with some of my mentors at Santa Barbara, Kevin Sullivan and David Asai, were really saying, you know, DNA is the way of the future in terms of where these kinds of interesting questions are going to lead. And so that then prompted me to apply to the Department of Molecular Biology rather than to the Department of Biochemistry, because naively I thought that there was more of that in molecular biology. It turns out that was going on in both departments, but that's what I was sort of looking for, was something where one could study things in a very mechanistic way and understand the DNA.
And so you decided to work under Professor Blackburn, and you became interested in her work and extending her work in telomeres. So help us understand what a telomere is. Yes.
Yeah, so it's the very end of the chromosome, and Liz had been working on this for a number of years, and it was something that I hadn't really heard very much about, so I was particularly interested in it because it was a little bit out of the mainstream. But they are the ends of chromosomes, the functional unit at the ends of chromosomes, which are essential for protecting the chromosome ends.
So it's like a little cap.
A little cap that protects the chromosome end. But it turns out that in addition to protecting the chromosome end, it's important for maintaining the length of the chromosome. And that was something that at the time Liz and others were recently aware of, that the way that when you copy a whole chromosome, it turns out that the way you copy a chromosome, the very end of that chromosome, the telomere, can't be completely copied to the end. And so there was this mystery about how it is that the end of the chromosome can be maintained. Why isn't it just that the telomeres get shorter and shorter and shorter every time a cell divides? That was certainly the prediction. And so we were interested in how that problem is overcome.
And as you begin this work and really extend it over time, you're, and I've learned this in interviewing the scientists who've come on board, you're actually on the back of people who have preceded you and made discoveries. And so let's just talk a little about that. So let me see if I have this here. In the 30s, what did Mueller and McClintock do that set the stage for what you were doing?
So the function of telomeres in terms of this protective cap, that's what H.J. Muller and Barbara McClintock showed, was that Muller was working in Drosophila and McClintock in corn or maize, showed that there must be something protective at the end, because when you break that chromosome end, that you get the chromosome is either degraded or it will fuse with other chromosomes. So they were the first to functionally define the telomere as we just described it. And then it was in the 1970s that Elizabeth Blackburn, working together with Joe Gall, showed the molecular characteristics of the telomere. So they defined the actual DNA sequence at the time. And then understanding that molecular characteristic, we could then go forward and ask questions about how the end of the chromosome is maintained.
And explain a little more what understanding the end and the molecular structure at the end, what is that about? In other words, it's help me understand that.
Yeah, so if you understand the detailed makeup of the chromosome, then that allows one to do experiments to be able to test how is it that the end of the chromosome can be maintained. But if you don't know what it functionally is made of, then you can't design experiments around that. And so it was really understanding that DNA sequence, and in the case of the telomere sequence that Liz Blackburn defined, it was from the ciliated protozoan tetrahymena, which has 40,000 chromosomes.
So you pick a one-cell organism that's doing a lot of this work.
Exactly. It's got 40,000 chromosomes, so you go to the source to find out what the the material is actually made of. And so they found that it was this very simple repeated sequence. And in DNA, we use the four-letter alphabet for the building blocks of the DNA, and that's T, C, A, and G. But what they found was that this telomere was made up of a very, very repetitive T, T, G, 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 T, T, G, 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 T, T, G, 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 hundreds of repeats. Very, very simple and repetitive. And that's what actually provides the protective cap at the end of the chromosome.
And does that mean that the cap is made up of individual caps, or it's one continuous structure?
It's one continuous structure, and then that DNA sequence actually has proteins that bind along it to carry out the function of the telomere.
And do different organisms have different lengths, and then throughout the aging process, what happens to these caps?
Well, at first it was only identified in these single-cell ciliates, and then as more people started doing experiments, once you know what an initial sequence is, then it gives you something in mind to look in other organisms. And so one of the very nice things about working on something as fundamental as the telomere is that things that are that fundamental inside cells tend to be conserved throughout organisms. And so how it's solved in one organism will tend to be true in many, many organisms. And it turned out that was very true for telomeres. So as people started identifying telomere sequences in different organisms, there were also these simple repeated sequences on the ends of chromosomes. And so we were able to then do more experiments to try and understand how those are maintained.
And Blackburn and her associate Gull, what part of their experimentation which really pushed this along was the discovery that you could take one organism and essentially build caps in another related organism, but a different organism.
So that was the experiment that had been done just before I came to Berkeley, and that was a collaboration of Liz Blackburn with Jack Shostak, and they took a piece of DNA from yeast, which is normally a circle, and then they opened that circle and added the telomeres that they had identified, that Liz had identified from tetrahymena. So instead of a circle, they now had what would be a linear chromosome, like most chromosomes. And quite remarkably, they found that when they took that linear piece of DNA with telomeres and put it inside yeast cells, and tetrahymena and yeast are in two different kingdoms of life, that function, that telomere function, was maintained inside the yeast. And so the telomeres functioned, and you now had this linear chromosome. And so that was really a key experiment. But even more importantly was then going and looking to see within those yeast cells that tetrahymena telomere had a little bit of telomere sequences added onto it that were yeast-specific sequences as it was dividing. And that provided the clue that made Liz and Jack suggest this mechanism. How is it that telomeres are actually maintained? And they made the very bold proposal that maybe there is an enzyme that actually will just add these repeated sequences onto the ends of chromosomes. So as I said, normally you would expect the telomeres to shorten progressively with each cell division, but they proposed that if you can then add something, then you can balance out the shortening with addition. And so that's what I then set out to test when I came to work with Liz Blackburn.
Well, just out of curiosity, is the sequence in the different organisms the same?
It's not exactly the same. It's the same flavor, that is, it's tandemly repeated sequences. But very conveniently, the yeast sequence is just slightly different than the tetrahymena sequence. So you could see that you had tetrahymena telomeres, and then there was a transition into yeast telomeres at the very end of the sequence, which allowed them then to propose that they could be added by an enzyme.
So, okay, you're a first-year graduate student. You've studied enough to know who you want to study with, and you have been exposed to the history that we've just talked about in terms of discovery. So then what did you say to yourself about what you wanted to focus on?
Well, for first-year graduate students, at least at the time, and I think this is still true, is you're required to do projects in 3 different laboratories. So I did a project in one laboratory, and then I did a project with Liz Blackburn on telomeres from an unrelated organism. And then I did a third project. And you're not officially allowed to make a deal with who you're going to work with until sometime in May of the first year. And I knew that I wanted to work with Liz during the time that I had done the rotation with her. And I knew about this idea, how do chromosomes replicate, how are they maintained? And I thought that that was the most exciting thing going on in telomeres. So in May, at the end of my third rotation, I made an appointment to go and see Liz, and I had two things that I wanted to ask her. One is, can I work with you? And two is, can I work on this project? And I was thrilled because she said yes to both. And the conversation lasted maybe 5 minutes. And then once I finished up my third project, I went back to Liz's lab and started the work to try and identify, is there such a thing which will add these telomeric sequences onto the ends of chromosomes?
And it sounds to me like, just following your intellectual journey here, that you were really, everything in your life came together to prepare you for this choice of what you wanted to work with and set about doing it.
It just seemed like the obvious thing to do because it was a very exciting problem that I was just curious about. But certainly that is built up by my experiences from the past that brought me to that point where I felt that this was an interesting project. And Liz was very interested in this, and she was happy to have somebody that wanted to pursue it.
So you start this work, and then what is involved in the experimentation that says, okay, what's going on here, and how do I play with these creatures, shall we say, or these chromosomes to figure out what's going on? Give us. You at some point I read you said, you need a strategy so what was the strategy you set upon, and what were the different things you had to adjust to come up?
So it's an empirical science, and we started, of course, again with tetrahymena. Tetrahymena has 40,000 chromosomes, and it must have a lot of whatever it is that is needed to maintain the chromosomes. So we used tetrahymena cells and basically, made a little cocktail of ground up tetrahymena cells with what we hoped would be the right pH and buffers and concentrations. But then the key was really trying to understand how we were going to look for something being elongated. And we tried a variety of different things. And we tried, isolating a specific piece of DNA that had a little bit of telomeric sequence on it. Did experiments for, 5 or 6 months with that, getting little hints here and there. But it was really, because it's an empirical science, it was just trying different things. And it was about 9 months after starting these experiments, where every day I'd come in and make a little cocktail of the tetrahymena cells and put them in with some radioactive DNA precursors to look if those precursors would be elongating something. And then instead of doing a DNA fragment, I switched to trying, well, maybe a synthetic DNA oligonucleotide would be the thing that could be elongated. And the very first time that I did that experiment, when I came in to develop the result of the experiment, I saw this pattern that looked like a 6-base repeating pattern of DNA. And the key in retrospect was in switching to using that DNA oligonucleotide, not because it was synthetic, but because of the amount that you could get in, because it was so short. It was 18 nucleotides long, 18 building blocks of the DNA, as opposed to something that was 1,000 or more. And because of that, there could be more molecules in the experiment, because it's so small that when you're adding a certain amount, the number of ends that were there was over 1,000-fold what we had been adding before. So it was just trying different things. And it was very clear as soon as we had enough of the material in the extract that there was this repeating pattern. But that's when things got interesting, because it was like, wow, this is exciting. There's something that we expect to see here. But then it's like, what could be fooling us? And there was a whole another year then of trying to figure out, maybe this was a known enzyme We were just interpreting it in a certain way. And we had to then convince ourselves that this really was something new. And this is something that I talk to students about often, is that when you have something that's new and exciting, rather than saying, oh, I want to find evidence to support my hypothesis, instead, we tried to bash down our own hypothesis and to find ways to knock it down and find out, how could this possibly be wrong? Because I would rather disprove my own crazy idea than have somebody else disprove it.
But we'll talk about what you had to do in the following year, but I just want to go back to this aha moment. And I believe I read that this was on Christmas Day that you came in, you see what has happened, and so this was your wow moment. And then what did you do? You went home and you were so excited and you did what we all do, played Bruce Springsteen.
Exactly. Because the pattern was something that was suggestive that this could really be, something very exciting. And then after that it was, well, what else could be fooling us here?
And I want to go back to this thing about the collegiality, because the new item that you were using that made the difference was actually something that you were able to borrow from a colleague who had been working on this?
Yes, there was somebody else in the lab, Eric Henderson at the time, that was working on actually the structure of these DNA oligonucleotides and doing nuclear magnetic resonance to try and understand. So he was just doing experiments just with this. And so he had it in the laboratory, and I said, well, can I have some of that to try in this elongation experiment?
So once you see this lengthening process, then you're then asking yourself, well, what is this enzyme and where is it coming from? Is that the next?
Is it really a new enzyme that is just adding onto the ends of chromosomes? Nobody had ever heard of anything like that before. Typically, if you have something that makes DNA, what it does is it has something to copy, what we call a template. So a typical DNA synthesis is there's a template there, and then the DNA polymerase, as we call it, the thing that makes the DNA, just copies what is there. And so we were wondering, maybe there's some small template somewhere that we don't know about, and really this is just a normal DNA polymerase that is making these copies. And so we then set out to devise a series of experiments to disprove or to rule out that it was just a DNA polymerase. And that took about a year's time. So the discovery was made in December of '84, and the paper was published in December of '85.
And so what then is involved in the experiments that followed in the next year? Seeing where this enzyme was coming from, or what made its work possible?
If we could rule out that there was some other template that was being copied. So purifying the enzyme, trying to get rid of any contaminating DNA that might be there. But what ended up really convincing us was really the converse of the experiment that Liz Blackburn and Jack Shostak had done. Because what they had done was to take tetrahymena telomeres and put them into a yeast cell and see that yeast sequences were added on. So what we decided to try was to make a synthetic telomere of the yeast sequence and put that into a tetrahymena extract. And sure enough, it was elongated, and the yeast sequence was the initial sequence, but what was added was the tetrahymena repeat. And,
which was the opposite of what had happened.
It was exactly the converse of the previous experiment. And because of the way that the sequences are set up, there was a slight shift in the banding pattern that we saw on the gel. And that was the second aha moment of this series of experiments that went into the first paper, because then I was really convinced that it wasn't just copying something that was already there, and that it probably was actually adding on to the end of the chromosome.
Now, when you say a shift in the banding, you mean explain that.
So the way, as I mentioned, they have this 6-base periodicity that was the 6 nucleotides that are added, TTA, GGG. Well, the yeast sequence, it turned out, didn't have 4 Gs in the initial template the initial primer that we were putting in. It only has 3 because that's the yeast sequence. And so the pattern then has to make 4 Gs. And so it adds one before then going into the rest of the pattern. So everything is shifted up by one in that, with that yeast sequence, which really fascinated us.
Now, the next step, as I understand it, and again, this is not my background, so I'm trying to get you here to explicate, and you're really doing a great job here. So when does RNA, the realization that RNA has something to do with this? Explain
so that was the next. Once we convinced ourselves that we really have a new enzyme that is actually taking an end and adding things onto it, then the question becomes, well, where does that information, how does it know how to make TT, GGGG, TT, GGGG? Because typically things do copy a template. And that's when we thought, well, perhaps the enzyme itself has within it a little template, and that perhaps could be made of RNA. And so that was the next thing we then set out to test is, could it be that this enzyme is what we call an RNP enzyme? It has both protein and RNA within it. And that was the next series of experiments that we identified something that looked like it might be an RNA template, but then we had to ask ourselves, can we really prove that it is the RNA template? And another series of experiments to come at it from a variety of different angles to ask, could we be being fooled just because you take the enzyme and treat with something that destroys RNase and activity goes away? Is that because there's an RNA template, or is that for some other reason? So it was really setting ourselves a series of challenges.
And what when you published your initial findings and then the ones that followed, how controversial was what you were saying for the field at that time? You said you had to be your own devil's advocate, so clearly you anticipated that people would be coming at you. And what did you anticipate?
I just wanted to be sure that we were right if we were going to go out on a limb and propose something like this. There was an alternative model whereby one could use a DNA recombination to get this elongation. So there were two models that were out there that were published, and we were looking for evidence for one of them. But it's very interesting because it didn't end up really being too controversial. People accepted it. We had to make some changes to the paper. But all along people said, well, you know, isn't that cute? That's a nice little thing that they do off in this organism, Tetrahymena. And there wasn't any realization that this might have some sort of a general role. And you know, now what we know about telomeres and telomerase, and they play a major role in human disease, and we had no concept of that back then. We were just trying to understand the fundamentals of how telomeres were maintained. We didn't even know that the human telomeric sequence was the same repeated kind of very simple telomeric repeats. So I wouldn't say that it was necessarily controversial, but we published in good journals, but it wasn't taken as a huge deal.
So your aha moment, in a way, wasn't a big aha for the community generally because it wasn't clear, well, so what?
For the telomere community, the group of maybe 40 labs across the world that worked on telomeres, it was a big deal. But I would say the molecular biology in general, telomeres weren't really on the radar. You know, there's some little problem over here with replication, but what's that got to do with, people were interested in understanding how genes are transcribed and regulation and development of organisms. So the fields that were really big deal fields were a little bit aside from what we were doing. So it was sort of on the side.
So in a way, we're learning here something about leadership in science, because here we have curious people building on a tradition, learning stuff from their colleagues that helps inform what they're doing, and in a way, opening up a whole new field. So initially, you're opening up the field for the people who study these telomeres, but now we go on to say, hey, this may have something to do with our understanding of cancer, cystic fibrosis, and other things. Talk a little about that next step.
Yeah, so there had been experiments or proposals back in the 1970s. So this idea about how when you replicate the end of a chromosome, you can't replicate to the very end. Liz Blackburn and I were aware of a publication that Jim Watson, the discoverer of DNA, had proposed, showing that you can't replicate to the very end of the chromosome. So we were citing his papers all along. Well, it turns out that around the same time, there was a Russian scientist, Alexei Alovnikov, who had independently come up with the same idea, but he didn't call the ends of the chromosomes telomeres, he called them margins. And so his paper was called A Theory of Marginotomy, so it wasn't pulled up by any of our references. But a colleague of mine, who I met while I was in graduate school, Cal Harley, who worked in the field of cellular senescence, was aware of this paper, and that this fellow Alovnikov had proposed not only do the telomere shorten, but that might play a role in the fact that when you take primary human cells and put them into culture, they go through a certain number of divisions and then they stop dividing. And that's known as the Hayflick limit, proposed by Len Hayflick in the 1960s, that there's a finite lifespan that normal human cells have. So in talking with Cal Harley, he made me aware of this other literature, which was going back to the 70s as well. And so then when the human telomeric sequence was identified, which wasn't until 1988 was the presentation at a conference that I was at Cold Spring Harbor, and I believe the publication was in 1989. Then once we knew what the sequence was, so really understanding what the components are is enabling. Mm-hmm. Once we understood that, then we could do an experiment to ask what does happen to human chromosomes. And so I collaborated with Cal Harley, and this is what's nice about science, is that you can do experiments in a blinded fashion. Because I really like to know what is the truth. And if you do a blinded experiment and you don't have any way of prejudging what the answer is, then you can really believe it's true. So Cal Harley had grown some of these human cells in culture for a number of divisions, and then he prepared the DNA and he sent it to me with just numbers on the tubes. I then took that DNA and was able to look at the length of the telomeres because I now knew the sequence of the human telomere. And so I did this experiment, and some were long and some were short, and some were long and some were short, and then I called him up and said, okay, what's the key? Mm-hmm. And sure enough, those cells that had been dividing for more times had shorter telomeres than the ones that, and again, that was very satisfying because I couldn't have been biased in any way because I didn't know when I did the experiment what the result would be.
So help us understand what the meaning, the possible meanings of this is for coming up with, say, drugs for cancer and so on.
Well, so what we were able to show is that this cellular senescence, that is when you take cells and they divide for a limited time in culture, that the telomeres would limit them from dividing more times. And one thought at the time, and now I think it's also there's support for it, is that the fact that normal cells only go through a finite number of divisions means that cancer cells have to have something wrong with them, and perhaps the telomeres might play a role in that. So the finding of the telomeres that shorten with normal cells in culture then led to the question of, well, what happens with cancer cells? Mm-hmm. Because the difference in a cancer cell is that it goes through many more rounds of divisions than it normally should. And so sure enough, again, with Cal Harley and Sylvia Becchetti, we were able to show that when the normal cell becomes a cancer cell, now this enzyme telomerase becomes active. And so telomerase is active in cancer cells and not as active in the normal cells.
So that if you could figure out a way to work on that process, then you might have a pathway to all kinds of discoveries.
So there were many thoughts and many papers published that if you had a way to inhibit telomerase, you could inhibit the growth of cancer cells. Now that we know a little bit more about the role of telomerase in normal cells, because it turns out it's not just cancer cells which have to divide many times. There are cells in your body which have to replenish other cells. One example, for instance, is the blood cells. Within the bone marrow, there are cells that normally divide every day to allow new blood cells to be born, because blood cells have a very limited lifespan. So there are normal cells in the human body which also have to have telomerase to be able to maintain their telomeres. So the initial story, which was a cancer story, we are very interested in that still, and we've done a lot of experiments to show that, in fact, limiting the telomere lengthening does limit the growth of the cancer cell. But if you're going to take that as a way to do some sort of therapeutics, you also now have to keep in mind the role of telomerase in normal cells.
I see. So how it was when you saw these other pathways and the general implication for medicine and disease and so on that you were then recognized with the Nobel Prize, because there was no Nobel Prize when you made the initial discovery.
Right. The initial discovery was just understanding the elongation of telomeres in tetrahymena. But then by continuing to follow our curiosity, I collaborated with Cal Harley on the senescence side of things, and then got interested in understanding the role of telomerase in cancer. We made a mouse that doesn't have any telomerase and looked at what happens and what are the consequences of not having telomerase and how does that affect cancer. By just continuing to be able to follow our curiosity, which is what's so nice about being able to work in academia, and the National Institutes of Health has supported my research for the last 30 years, and if I can write a grant that's interesting enough, then I can follow and do those things. And so by following those things up, the consequences then for human health became clear. And the thing, as you're saying about the Nobel Prize, is that it's given for physiology or medicine. And so the medical implications of the telomerase and the role in cancer and in stem cells became apparent. And then what the Swedish Academy looks at is then, okay, who was it that made the discovery that allowed this to be clear?
In an interview you gave when you won the Nobel Prize, you made a point that I found very interesting, which is that it was very important for you to bring your children to your first press conference. Talk a little about that, because you make the point that men don't do that when they win the Nobel Prize, or they hadn't done it.
Oh, I forgot that I had made that point, but certainly I wanted my children to be there. I wanted them to share the day with me. Mostly that's how I felt, that it was a family event. And so when I got the call at 5 o'clock in the morning, and then after the phone call with the Swedish Academy, I went and I woke up my children and I said, by the way, I won the Nobel Prize. And the first thing my son said was, "Do I have to go to school today?" And I said, "No, you don't have to go to school today."
Now what I want, the reason I wanted to pursue this is it's really about it's saying something about women in science as opposed to men, and I wanted to talk a little about that. So in this work, obviously, two women are very important in the work, and we're grappling with the issue as a nation of how do we get more women into science. Talk a little about what women bring and how to scientific discovery. I mean, you're still doing the same processes. It's not as if you're inventing, a new science. But talk a little about why we need more women and what they bring to the table.
Well, I mean, the fact that that there's only about 30% at the assistant professor level, 30%, and even higher at at higher ranks of women in science, really is just unfortunate in terms of the amount of brain power that we have in the United States. If we're going to have 50% of the people that may have creative ideas not really be able to engage, then that certainly takes away a lot of the possibilities for discoveries. So I certainly don't think that women necessarily have anything unique to offer for the scientific enterprise, but they have a lot to offer. And a lot of times the way our institutions are structured, women aren't included in that process. And so science is to the detriment because of that.
And what can we do about those structures? What in particular holds women back?
Yes, I think that that's a really a larger issue than just women in science. That really has more to do with our society in general, because in society at large there are discussions ongoing about women at higher positions in business, on corporate boards, on various institutions like that. And so I don't think that science is unique in this area. That as a society, we have certain norms about how men are perceived and what kinds of jobs they should have, and how women are perceived. And although a great amount of change has occurred, for instance, since my mother's generation, really the numbers haven't followed suit. For instance, when I was at Berkeley in graduate school, 50% of us were women in graduate school, but that has not tracked along. You would expect then, if it were, as people have said, it's a pipeline issue. There aren't enough women coming into the sciences. If it were a pipeline issue, we would be at 50% at the level of full professors now, and we're at the 10% or so level. It's not a pipeline issue. I'm certainly not an expert in what are the causes, but I've read a number of reports. There was a very nice report by the Association of University Women that suggested that it's a leaky pipeline. That although there are 50% women coming in, that at the different transitions from being a graduate student to being a postdoctoral fellow, being a postdoctoral fellow to an assistant professor, at each stage, more and more women decide not to go on in the pipeline. So they're coming out of the pipeline. And the question is, why? And what are the structures that we have in place in science, but also in general in society, that more women make those choices than men make those choices.
And you're suggesting that it may be a larger structural issue that applies to the society in general.
I certainly, when I listen to national fora on these kinds of topics, the issues that I hear for women in banking and women in business are very similar. And actually, I feel like the women in science probably have a little bit of an advantage because if I were a banker today, I would have to be certain hours at work, and I do have 2 children, and so it's nice for me to not have to be quite so proscripted in how many hours I'm at work. If I have to go off and see my son in a play or something like that, I can do that. I have a lot more flexibility as an academic scientist than I would in other areas. But yet, still, the same kinds of issues of this leaky pipeline exist across the country. I was very interested when Anne Marie Slaughter, who was at Princeton and was working with Hillary Clinton in the State Department, came out with a very provocative paper in The Atlantic on why women still can't have it all. And clearly, even in her field, in political science, there's very, very similar issues that prevent women from advancing.
If we look back at your intellectual journey, you were blessed with many informal mechanisms, informal environments that made you the great scientist that you became. I want to ask you about science education. I mean, you were blessed with a family environment in which you breathed science, even if you didn't decide you wanted to be a physicist. At Santa Barbara, it was a setting which was not formal and that would draw on your strengths and really prepare you for science. When you came here, you were lucky enough to work with a distinguished woman scientist, and that might have been sort of conducive. So do you have any thoughts on what your journey tells us about what we might do with regard to science education generally?
Well, the one thing that was very clear that helped me a lot was being able to try out different things, because sometimes you can't just read about something and say, yes, that's what I want to do. To find out what fits intellectually, sometimes you actually have to be in the situation. So that's something I definitely encourage all young scientists or young people in any area is to go out and experience a lot of different things. Because when you're in the environment, you can then see what captures and what fits best with your intellectual style. And then you can know what it is that you want to pursue. So I think that having those kinds of opportunities in a number of different settings was really very, very helpful to me.
And sort of looking to the future of the work that you do, what vistas do you see in terms of questions that we have to answer along the path that you have helped define?
Well, this issue of telomeres and cellular senescence has really become much more focused on age-related disease and the role of telomeres in age-related disease. I mentioned the blood cells, so bone marrow requiring telomeres. And it turns out that there are a number of age-related diseases like immune senescence, bone marrow failure, something called pulmonary fibrosis, liver fibrosis. So there are these diseases that tend to be diseases of aging where the short telomeres clearly play a causal role in those diseases. And so we're even more interested now in understanding the detailed mechanisms of how telomeres are maintained. And despite everything that we've done for the past 25 years, there's still a lot that we don't understand about the regulation of this process of their telomere elongation and how does it know how much to elongate. And if it doesn't elongate enough, then people are at risk for these age-related diseases. So really understanding that, and we have to know more of the actual components because that's an enabling kind of discovery. So there are still many open-ended questions about both the detailed mechanism and then how that relates to disease. Why are there these particular age-related diseases that are sensitive to telomeres and not others? So I really think that there's a huge amount still to be done in this area.
Well, on that note, Professor Greider, I want to thank you very much for coming back to Berkeley to deliver the Hitchcock Lecture and taking the time to be on our program. Thank you.
It was my pleasure. Thank you.
And thank you very much for joining us for this conversation with history.